So, hi guys, for the fifth session of What Geochemistry Tells Us. Today, our first presenter is Serban Sarbu. He's a researcher at the Mirakovic Institute of Speleology from Romania, but also at the University of Chico, California. So, Serban, we have a presentation of stable isotopes and how we use it in different subsurface cave environments. Very interesting one. So, Serban, it's all yours.
Okay, can you let me share the screen?
Yeah, you can share it.
Let's see, yes, okay, good. So, okay, I'm going to talk about the use of stable isotopes in the study of caves which contain some reduced chemical compounds, mostly hydrogen sulfide, but also methane and ammonia. Of course, other reduced compounds can be present, and they are used by microbes because microbes are really good at using anything they can. But mainly the ones that I will be mentioning will be the sulfide oxidizers and the methane oxidizers. Now, there are very few caves in the world that qualify as caves that contain chemoautotrophically-based ecosystems. The vast majority of caves are just the regular ones where there's not much food, it's dark, there's no photosynthesis, there's no food. But that makes them similar to the deep sea environment. However, when you have a source of food, such as the presence of some sulfidic springs or springs that contain methane, both on the bottom of the ocean and in caves, all of a sudden you do have a source of food. And that's really what my research was concentrating on. I dealt with the study of such ecosystems, starting with 1986 when Movila Cave was discovered, and then following this pattern in other caves around the world. I will start with a few slides, and I apologize to those who are very familiar with this type of science, but I just wanted to mention a few words about stable isotopes. They are these forms of a certain element that are placed in the same box in the periodic table, hence isos, same, and topos, place. The most known are probably the hydrogen isotopes. When you add a neutron to that one proton, it becomes deuterium, and if you add two neutrons, it becomes tritium, which is a radioactive one. Oxygen also has several isotopes. The most common is 16, but there is also some 18, and I apologize, there's a mistake here. That should be 17. Carbon has 8 isotopes. They range from 8 to 16, and nitrogen, the most known are nitrogen 14 and 15. One of the most common and most known is carbon 14 because it's used for dating, but we are not for age dating, that is. We are not going to talk about the radioactive ones, but about the stable ones, carbon 12, carbon 13. And then sulfur is another one that we are interested in cave studies. There are certain materials that I used as standards, and I list them here. The other thing I wanted to mention is the delta value. Now, this is a way of expressing the stable isotope ratios, but it's done so in order to have some numbers which are easy to follow. So If you look at 1.0010 compared to 0.9990, it's kind of hard to figure out what's going on. But when you change it to a delta value, you see the difference between 1 and -1. So that's easier to understand and to follow. There's a difference between heavy and light isotopic values. What does that mean? For example, in carbon-13, in a delta value of carbon-13, when we look at green plants that live at the surface on a meadow, in a field, anywhere, the stable isotope value of that grass, of those plants, would be about -27 per mil, just to give you an idea. However, what does it mean when we look at light stable isotope values? Let's look at values of fauna that we collected in Mobile, much lighter than -27. So it's -42. It means that there is less of the carbon-13 here compared to here. So these are just some introduction slides to give you an overview of the notions that I will be using in my presentation. Now, what is fractionation? If you are an enzyme, you'd learn very fast that if you have to do work with some atoms that are lighter and heavier, and if you have to do this 24/7, every day, all day, you will learn very fast to select for the lighter ones because it's less work for you. So fractionation is kind of functions kind of in this way. The enzymes would prefer the light isotopes. So at the end of the process, we obtain a product that's isotopically lighter than the source. It also applies to evaporation. If you have a pond, the molecules of water that will evaporate first will be the lighter ones compared to the heavy ones. So the lake will be enriched in heavy isotopes as time passes. Now, in food web analysis is where I use the stable isotopes to help me figure out what's going on in these caves. The rule is you are what you eat plus 1.5 per mil, which means that green plants are, let's say, 28 per mil. Now, a rabbit comes and eats those plants. The rabbit will use the green grass in respiration, thus it will burn first the light CO2. What remains is heavier, so the rabbit will be heavier. The wolf who eats the rabbit will be even heavier, the fleas on the wolf, the parasites on the fleas, and so on. So we get to lighter, I apologize. So we start with heavy, we end up with light. That's basically, I apologize, I got it wrong. So we start with heavy grass, we go to lighter, excuse me. We'll see that on the following graph. So the grass is isotopically 27, rabbit is heavier here, wolf is even heavier, fleas are heavier, parasites are heavier. Now, in photosynthesis, the process goes the other way around. The enzyme would select for the light carbon isotopes. So if we start with CO2 about -7, as you see here, the organic molecules will be much, much lighter from the point of view of stable isotope values. So let's go now to what I've been doing. And again, I apologize to those who are very familiar with these notions for my general introduction. This is a satellite view of the area around Mangalia in southeastern Romania. This is a swamp that has several spots that you see here in white. In those swamps, there are very strong springs that push sulfidic water that comes from the depth of about 200 meters, where it's stored in an artesian aquifer. Along geological faults, this water reaches the surface. We were looking for places in Romania where there's a potential to find long underwater caves. And this was one of the places we're actively looking at. Now, in June of '86, a geological shaft was dug to investigate the geology of this area, and this is the crew that eventually found and later protected Movile Cave. It starts with this artificial shaft, which is about 20 meters deep. We have to descend using ropes, or in this case, electron ladders. And eventually then the passages are rather small. We crawl or have to duck to get through. And eventually we reach this lake, which contains sulfidic water. The interesting thing for me as a biologist was the fact that there is a lot of fauna around this lake. Now, for a geologist, you'll notice that the walls are heavily corroded, and that's sulfuric acid speleogenesis. Hydrogen sulfide from the lake would evaporate and then dissolve in the moisture on the walls. What results is sulfuric acid, which dissolves the rock very fast. So we have this Swiss cheese look of the rocks in this cave. My interest was not in the speleogenesis, but mainly in the structure of the food web in this cave, trying to understand why so many animals are present, so many species of invertebrates. What do they eat? Why are they there? So we found large numbers of interesting cave-adapted species, but also huge numbers of individuals. This is one of the most beautiful ones. It's a pill bug or wood lice, that's how the British would call them, or terrestrial isopods. This is a centipede, very active, large, about 10 centimeters long. It's a new species as well. Pretty much everything in this cave is a new species and endemic to the cave. We have 37 endemic species for this ecosystem. This is another interesting one, a water scorpion that is blind. It's the only species of hemipteran that is cave adapted. Then here is a map of Movile Cave. The upper level that you see here in the profile view, and this is a schematic view of the cave, is dry. There is not really much there to find in terms of biology. This looks pretty much like any other cave in the world. No food, no animals. Interesting from the point of view of geology, mineralogy, and so on, but not biologically interesting. However, once you get to the lower level, the lake, and then you cave dive to reach AFL 1 and AFL 2, that's where biology is present, and there's a lot to see. When we explored the cave and first surfaced in these AFLs, we noticed this floating biofilm that covers the surface of the water. Of course, we didn't know at the beginning what it was, and it took us a while to figure it out. But eventually we realized these are microbial mats that are kept afloat by bubbles of CO2 and methane that are keeping them at the surface. These squares we mounted in order to determine how much biomass is produced in 1 month. So we would harvest this surface of 10 by 10 centimeters. But still, it was very difficult for us to figure out what do these microbes do. So what we tried was to do incubations with radio-labeled bicarbonate. We used radioactive carbonate, bicarbonate at that point. And then we did some readings to see if this radioactivity was incorporated by the microbes. And the results were good, but not very, very good. And then we got in touch with our colleagues who studied and still study the deep sea vent ecosystems. And these were discovered about 9 years before Movile Cave. And those people spent a lot of money and did a lot of good work on these ecosystems at the bottom of the ocean. One of the methods they used, and that's what we borrowed as an idea from them, was the use of stable isotopes to determine what is the source of the carbon. So here we go, we have a graph of what we found in Movila. This was information that eventually gave us a publication in the Journal of Science in '96. And a few important values to remember. Limestone, which is in fact the standard for carbon, has a delta value of 0. Now, CO2 in the atmospheric air outside the cave is -7, green plants outside -28, methane -60, and Movila Cave fauna -45. Let's have a quick look at this part of the graph. So let's consider these are aquatic, these are terrestrials. The other one with M, those are the marine samples that we analyzed from the Mangalia area. And then all of this here is from Movila Cave. Now, if let's think of this as being a producer, one of the green plants. Now, if somebody would come, if a grazer would come and eat this plant, the stable isotope value of that grazer would be somewhere up in this direction, moving this way toward isotopically heavier, because the light CO2 was pushed out by respiration. So now a predator comes and eats that grazer, that predator would be here, and so on. So we would go from here in that direction. Now, when we analyzed these samples from Movila Cave, we realized that there is absolutely no way that the green plants from here would be the source of food for the subterranean ecosystem. There's definitely something else going on. There's definitely another source of food that these creatures rely on. There's no way that food can be the food that these creatures rely upon. So now trying to figure out what this origin of the food was quite a challenge. And eventually what we realized was that the water in Movila Cave contains some methane, and the stable isotope, the delta value of methane for carbon is -60. So when that methane is oxidized by methane oxidizing microbes, what would result is light bicarbonate. And if that light bicarbonate is used by the sulfur oxidizers, the organic matter resulting from that process would be also somewhere here. So what we see here is really the influence of the presence of methane, both used as an energy source and used as a carbon source for these subterranean ecosystems. So this is really how we managed to show, and without any doubt, that this food is not coming from the surface, it's produced in situ in the cave. So this is a chemoautotrophically-based ecosystem. Now, later, we realized that there are several such places. Initially, Movila was the star of our research. We thought it's unique. There's nothing else like that on the surface of the Earth, but then, I should say, under the surface of the Earth, of course. And then we realized that wherever you have such conditions, if that's Frasasi caves, where the lower level is sulfidic, or if it's Melisotripa cave in Greece, where again we have a sulfidic lake, or the Ainur cave at Tabga in Israel, or Ayalon cave as well, and much more recently the Tashan cave in Iran. So in all of these situations, whenever sulfidic waters are present, there will be microbes present that take advantage of these sulfidic waters and produce food in situ by chemoautotrophy. Here is a picture of the Frasasi caves. You see, it's a huge system, absolutely huge. I think it's about 30 kilometers of cave passage, if I remember well. But the only sulfidic part here is not as spread. This is all fossil galleries. This is the Sala Ancona, 100 by 100 by 100 meters, all dug by microbes through sulfuric acid speleogenesis. It's a huge hole. If you talk about 1 million cubic meters of rock missing in that mountain. But the sulfidic system is only in this area. And in those areas, we found new species, endemic species, microbes that produce food and so on. And again, here we use stable isotope data to show that this food is not coming from the surface, but it's produced within the cave. Look at these microbial biofilms that are present in the water. For a trained eye, you would also see some of the amphipods here, but hard to see in this picture. Ramo sulfureo, the deepest and sulfidic section of the cave, again, floating microbial mats on the surface of the water here. It's hard to reach these places. It's really muddy. You can see me there back 25 years ago. But from the point of view of science and biology, it's really interesting, as well as from the point of view of microbiology and mineralogy, geology of the systems, paleogenesis, and so on. Here's a picture of Tabqa. It's a famous place on the Sea of Galilee in Israel. Famous because apparently that's the Bible says that that's where Jesus did the miracle of the fish, multiplication of fish and bread. As a result of that, there are many monasteries right in that spot, and under one of these there's a cave. It's a spring that connects to a cave. In that spring and cave, this creature was found. It's Typhlocaris Galilei. In fact, it's the largest cave-adapted invertebrate, about 10 centimeters long here. Beautiful animal, very elegant, long antennae, completely blind. Was discovered and described in 1912. And then the cave was studied in 1968 by a bunch of biologists from the University in Jerusalem. Interestingly, they had discovered back then the first chemoautotrophically-based cave ecosystem, but they didn't realize what it represents. So they didn't have enough background knowledge. We were lucky to find Movila Cave 9 years after the deep sea vents were discovered, so the homework was done for us. Here is Ayalon Cave in Israel, again, and also new species, endemic species here, much bigger cave, including, it contains also this scorpion, the only cave-adapted scorpion known. Unfortunately, only exoskeletons were found, no living specimens. And this is Cueva de Villaluz in Mexico, where the biology is very interesting, especially in terms of microbiology. You see the milky sulfidic water that flows through this cave. There's a lot of bacteria, but the cave contains big windows in the ceiling, so there's a lot of input from the surface, and it's hard to tell which is which. Let me switch to the second, well, I have a couple more slides. In fact, this is Melisotripa cave in Greece, recently discovered. All these dots that you see here are cave amphipods. The density of cave amphipods in this cave is about 200 specimens per square meter, which is unheard of in any other cave. Caves are poor in terms of food, poor in terms of fauna. The fact that we have food being produced right here in the cave allows for such high densities of species and specimens. Let me switch to the second part, which is now talking about gas-gas, oxic-anoxic interfaces in caves. The first one we studied was Sulfur Cave in Transylvania and Romania, central Romania. And we use smoke to visualize the interface between the volcanic gases emitted by this cave, about 2,000 cubic meters a day, and the atmospheric air on top, which contains oxygen. So we can safely stay in there upright as long as our head is above this interface, we're fine. There are other caves that we know of. We started their study, one of them in Tuscany, the other one in close to Naples, Grotta del Cane, the famous Grotta del Cane, and then some caves in Greece, which we hopefully will visit soon. This is Sulfur Cave in Romania. Please note, this is where the interface is located. Everything below it is 96% CO2, a little bit of hydrogen sulfide and methane. But above this yellow line, there is breathable atmospheric air. If we want to go in the deep section of the cave, we go in there using this scuba tank, which is hidden in this little backpack, and then a regulator, and we use a mask in order to be able to stay alive and not suffer the pain of walking in there without a mask when your eyes hurt and your nose hurts. So see here, everything below the interface is yellow, covered by sulfur. Everything above it is volcanic rock, some of it degraded. There's a lot of sulfate in there, various minerals, but right at 5 centimeters wide, we do have microbial mats, and that's what we were interested in. Myself, especially as I was looking for other systems, not only the water, air, as in Mobile, but also gas-gas interfaces such as here. Very interesting things we found there, such as, please have a look at this SEM picture of the microbial mats. These are some micro fibers that are used by these microbes to transport electrons up and down between the area which has oxygen and the area which is rich in hydrogen sulfide. The CO2 delta value in this cave is about 3.2 per mil. However, the biomass is minus 31, which tells us that it's not the sulfur oxidizers that produce the main portion of the food in this cave, but it is the methane oxidizers. So there's a little bit of methane in this cave. They are using that, hence they produce this light organic matter, light food for the rest of the microbial community. Interesting for the geologists, of course, is the presence of these sulfur deposits. This is an optical picture taken just with the camera, a macro lens. You see the layers of sulfur, how they are, we assume they are, this is deposited in winter and this is in the summer, but we don't know. This is some research that is ongoing. You saw the stable isotope value of sulfur here. The next steps will be to try to analyze the stable isotope value for sulfur in the emissions and the hydrogen sulfide. Look at these fibers, how they are very compact in some layers, like here, and then loose here. We assume this happens in the summer when the drier air allows for the drying of the surface, and then these fibers are pulled together in these bundles. The tips of these sulfur fibers that are pointed toward the inside of the cave, we don't know if microbes are involved in this sulfur deposition or not. This is ongoing research. Here is a picture from Bossoletto Cave in Italy. Due to the pandemic, of course, we couldn't travel there to take samples and measure anything, but our Italian colleagues were there using breathing apparatus, and they went in, they took these pictures, collected some samples which are being studied at the moment. We also see sulfur stratification, but so far we don't have any data on the microbes. That's ongoing. Anyways, I'm getting close here to the end of my presentation, the famous Grotta del Cane. Now, this cave, in fact, was dug by hand by Romans as a sauna, or what they called the den, a sudario. People would descend along this gallery and then spend time in a room which is right there in the back. In the meantime, the volcanism has changed there, and the cave got filled with high concentrations of CO2, so it's deadly to go in there right now. The temperature is about 55 Celsius. You cannot go in there without special protective equipment and breathing apparatus. This is the entrance of the cave nowadays. It's closed. People cannot go in there unless they have a special permit. But it used to be famous about 100, 150 years ago when it was part of the Grand Tour of Europe. So whoever wanted to see interesting places, they would stop by at Grotta del Cane. And there was a guy here who had a dog that he would bring into the cave, lower it into the lower section under the interface. The dog would faint, hopefully not to die, and he would then run to a little pond next door, throw it in the water, and the dog hopefully would wake up. And these tourists here would pay the guy some money. So it was a show cave in some very weird and macabre type of fashion. But basically, it was visited by Mark Twain, who wrote about it, by Goethe, by the author of The Three Musketeers, by Dumas, and I think Mozart was there. So it's a very famous place. We went there, we took samples, and they are being analyzed at the moment. So to conclude my presentation here, we use stable isotopes in caves, especially for food web analysis, but they are also being used in paleontological research, hydrogeological research to see what the origin of water is in caves, and then, of course, mineralogy and geology to determine the origin of the minerals and what's going on with the geology of areas where caves were found. So with this, I conclude my presentation, and I'm open to questions if there's time for that. Thank you very much for your attention.
Thanks, Serban. Are there any questions for Serban? So as a comment to Serban's presentation, when I first started to work with him, I didn't know you can use stable isotopes in biology or microbiology. So it was totally new. Wait, does the geologist that use it? No, we also use it, the microbiologist. So as working with Serban since 2016, 2017, he was always doing, okay, if you go to a cave or go to a volcanic region, open your eyes if you see caves or you see like holes with CO2 or you see sulfur, this oxic anoxic layers. So this is how we started to think about different things. Where could these systems be? This is how, for example, the new appeared one is Sosaki cave, a possible new appeared one.
So basically, I'm hoping that once you heard the story, you will also keep your eye open when you see any fumaroles that smell like hydrogen sulfide, or if you know there is any methane in those emissions, we will find microbes there. So there's absolutely sure. The rule is microbes in microbiology, the rule is microbes where everything is everywhere the environment selects. So if there are the right conditions, we will definitely find some interesting microbiology in these fumaroles. So please keep your eyes open. Don't. It's really funny when most people walk by a place that smells like hydrogen sulfide. They all go like, "Oh, this smells horrible." When I walk by a place that smells like hydrogen sulfide, I would just get very excited because I hope to find a cave that's interesting biologically. So it depends on the point of view, depends on where you come from.
So if there's no other questions, thank you, Chef, for the presentation.
Okay, sure.
And it was really nice to see you and hear you.